Always forget that. And share my screen. This time I'll not forget. And okay, so, but you can do these things using R, but sometimes it's taken too much time writing code and stuff. So what we are doing, we created this interface called D browser. So after you install D browser, you can install D browser to your own computers too. Like if you search D browser, like bioconductor D browser, whatever. So there's this bioconductor project here. So basically you need to run these lines for installation into your computer. If you install R or RStudio, if you run these, you're going to have RStudio in your own computer. It's going to be faster here. We have RStudio installed here too, the browser umassmed.edu. This is working here too, but multiple people are using here, so sometimes it might be slower. That's why some running on your own computers will be faster. So today I'm going to run it in my computer. So basically what you're doing is this. You're going to load the library, the browser, and hopefully it's going to load. And then after you load, you're going to start it with like this, start the browser. So what you're doing here is you need to upload your count data file. So how you get it? If you process your data yourself, you create a table. Or if you process your data in Dolphin Next, you can go to one of your RNA-seq runs and go here, for example, and go to the report section. So for the files you have, you will have the tables, and then there's download button here. So basically you're just going to download this and upload that file into your into here. So make sure the file is like the separator in the file. Usually when you download them from Dolphin Next, it's tab separated file. But if you create yourself, it could be comma separated or semicolon, whatever you put in between the columns, you need to use that tab. So if there's a problem in your file, sometimes there's even one NA or something, one empty like cell in your table, it's going to give an error. So it has to be complete table and matrix. So there are some tutorials here. So your table should be something like this. So you have your gene names, and on the columns you have your samples, and these are the count values. Even they are not integer in your file, we convert them to the integer. So in this case, these values are coming from RSAM. I told you before, these values calculated using a likelihood function. That's why these are not integer, but they're supposed to be integer. So, and these are like expected count table produced by RSAM is a row count file. So you're going to use that file. Don't use TPM file while you're uploading here. So another thing you could do is you can define your metadata file. This simplify your job drastically, because sometimes you have many samples. Like imagine you have 20 samples here, you have 5 different conditions. You can define those conditions. If you have batch effect, you can define your batch effect. If you have done the experiments in multiple like times or using multiple kits, so you need to do a batch effect correction, or you have to check at least you have a batch effect problem in your data. I'll show how. And then you can, if you upload this, you can do this kind of corrections in your data. The other thing that you need to know here is make sure you prepare like there are a couple of things you can do in Dolphin Next, in the browser that I'll show now. You can define multiple conditions even in the same field here, and then choose them which one you want to compare, and you can do multiple comparisons. Or you can define multiple columns here. the browser reads all the columns, whatever you define. So you can define the treatment or some other conditions, covariates in different columns here. And so let's come here. So I have a demo here. You can, if you want to play, you can press this button to load the demo. This is the same demo that you did your homework, you did in your homework. So we download that data and create it, like run the pipeline, and you can either upload here or you can just press this. So after you load the file, you come to the screen just to see if everything is right. You know, if there's no problem in your file. One more thing, actually, I forgot to tell here. So these names, like the sample names, has to match, exactly match with your sample names in your metadata file while defining the conditions, treatments, and batch effects. So if there is anything wrong here, for example, I see sometimes people are using underscore here, dash here, for example, they forget. So make sure, you know, you copy and paste, copy, transpose in Excel and then paste like that, however you want, but make sure these names are matching. Otherwise, you're going to get an error. I see lots of people they stuck in that, like just uploading file. It's because, you know, you have to be really careful about the file, complete count file, and matching metadata file. So after you upload, if everything is fine, you're going to see all the columns you defined in your metadata file. And also you're going to see the read counts. So you can tell if anything is wrong by just looking, because you already run this before and you know the read counts. And then after you filtering is actually, this step is, we are doing this in our bootcamp too. We did. So we are eliminating zero counts here using this filtering. We have a couple of methods here, like max, mean, and CPM. This is max, mean, almost the same thing. So it means that in all the samples in your data, so if, for example, max is this, so if in this line, if maximum read is less than 10, we are removing that gene. Okay. So CPM is count per million, and you can choose like it's normalizing, like count per million. If it is less than 1 in 5 samples, it's removing whole that gene. So for example, if, like, look at this sample, for example, if the calculation, I don't know, like now 1410 is going like this, but imagine if you normalize this with count per million, the equation is there in the help section. So if this is less than 1, it's going to remove this gene. Okay, so means here it's going to remove all these low counts here. And then after filtering, I'm just going to keep it in max and 10 filter. So we did this actually in our bootcamp. So we just remove this. After removing those zero counts, you have this distribution. There's still, like, this is not perfect distribution here, around like 3.1 or something. This is log 10. So, but maybe these are like, some of them here on this side is close to 0 because you see this bimodal distribution, but still it's okay. Like, you know, putting, you cannot eliminate everything because some of them are really low abundant genes. Some of them are like not maybe 0, but you know, you have to be okay with those kind of changes. Okay, so there is this batch effect correction here. So there are like lots of buttons here you can press, like show data, just showing the data, like how many lines you have. For example, at the beginning, we have 30,000 entries, 30,000 genes. And after we finish, there are 14,000 genes. See, like we eliminate 16,000 genes. It's extremely critical to do DE analysis because your p-adjusted values are going to take into account the number of genes you tested. So here, there's this button, batch effect correction. So if you have a batch effect problem here, like, you know, in this case, I don't have any batch effect correction, but any problem here. But so if you have, for example, if you're working on time series analysis coming from different donors, if donors coming from the same time are not like clustered together in this plot, then you need to do this batch effect correction. So you understand if you have it or not by just looking this PCA plot. And there are other plots we have here, like, you know, interquartile. This is before batch effect, after batch effect, or you can say that the distribution of the data, you can see if there is something wrong. Here, they can be like in different parts. So after correction, they almost come on top of each other. So you can test if the data is corrected or not. I'm just going to do it. Nothing is going to change, but when you're doing it, you need to use, like before batch effect correction, it requires to use a normalization method. So these normalization, mRNA normalization is coming from DE-seq2. So DE-seq2 intrinsically normalize the data by itself. That's why we need to give like row counts, because if you give normalized counts, it's going to be double normalization too. So you have to be sure that, but in this case, we are not going to do now we are going to do the correction first. If there's a real issue, by the correction issue, if there is not a correction problem, then don't do it. And then you need to choose the correction methods. We have combat-seq, Harman. So these are little different methods, but you can try each one. I'm not going to go into detail. There are papers for each. You can search and read the details, but basically it's just correcting the data when you define the batch. The one thing that you need to be sure, so when you have two batches, so in one batch you have only controls, in the second batch you have only treatment. So if you do batch effect correction, it is not going to work like that. Okay, you have to be careful. You have to have at least a couple controls, or one control and one treatment in each batch. So imagine, like, you need to increase the number of samples, but you think that you only need the like control, for example. Don't do just control. You need to add some treatments there too. Otherwise, you cannot do the batch effect correction, you know, because what it is doing, it is using all the treatments together, all the controls together coming from different batches, and adjusting the medians, for example. So such kind of normalization it is doing. So make sure that you have in all the batches you can have multiple batches, by the way, maybe 3, 4. So in each batch, you have to have every single condition you want to correct. Otherwise, it's not going to work. So then after you define those batch columns in your metadata file, they're going to appear here. And then you're going to choose your treatment and batch fields here, or treatment is condition, or whatever, like those correction you are going to make on. And then when you submit this, it will do the batch effect correction. And then you can see what happened, your data. Like, for example, you can color them with the treatment. See, these three are together, these three are together. So it's doing something, but this is not real batch effect. I just choose like the farthest point here, but there is no batch effect problem here, but still it is doing some correction. You can tell that it was 61% in PC1, explaining like the variance in these two different conditions. In this case, 64. So it is doing something interesting here that you can tell. There are other things you can do. You can put the controls here, like experiments here. You can put the sample names on it, and it's not perfect, but at least you can see what they are. And what else you can do? You can change, if you have like multiple conditions, not two, but maybe more than two, you can use the shape and different shape, and you can give different shape for different conditions. I have only two here. Okay, so after you do the batch effect correction, there are QC plots. I'm going to show you after DE, but let me do the DE now, that I'll show you the QC later. So in DE part, if you choose the select meta, so I told you, you can define many columns. To simplify this, choosing the columns here, you can actually use this. It's just changing the columns according to what you select, what you define in your metadata, or you can just define it here. You can remove. For example, in this case, I want to compare 2 samples, just to show you like what's the difference between 2 sample comparison versus 3 sample comparison. So you can add as many comparisons as you want. If you have like covariates in the future, you might have different conditions controlled by this third condition, for example, or the sex of the samples, like you would just want to correct something, then you need to define those covariates here by selecting that covariate column. Okay, for example, you need to add more metadata in. I told you, like, metadata is so important that you need to keep track whatever you have, and then using that metadata you can utilize and correct your data to find the best difference between 2 different conditions. Imagine, like, you think that female male like they will affect some calculations. If you think that you have that problem, then you can add this as a covariate to remove the effect of that field. And then when you can add multiple fields too. So when you add those covariates, then it's not gonna take into account that contribution of sex into your calculations. So in this case, like, there are a couple of methods here: DESeq, AJAR, Lima. I'm going to use DESeq too, but there are also AJAR for example, if your data is normalized, so you cannot use DESeq too. You can use AJAR and another test in AJAR. Like, you cannot use, for example, you can exact test, but you can use this GLM LRT. So for AJAR, or there's Lima, microarray data come always normalized. You can use this like Lima if you are working on microarray data. So, but in this case, we are using count data. I'm going to use parametric tests. These are the defaults. I'm not going to change anything. And the tests, I usually use wall test, but it performs better. And I use shrinkage. This is just shrink the data. When you do your GACA analysis, for example, for functional analysis like Go term analysis, when sometimes you need to have your list sorted using a statistics method called Wald statistics, for example, or another LRT is fine too. But this is working best for me when I use that GACA Go term or functional analysis programs, because you can sort it using that test. And I'll tell you more a little bit. So, and I choose my conditions, controls, 3 experiments, and I choose 2 by 2 here. And the good thing here, you can choose multiple comparisons, and at the background, when you go to your R, it is actually running that data. This is real data. It takes like a couple seconds, like 10 to, in one run takes 10 seconds, and then it's going to be ready. I did 2 runs now. One is for 2, the other one was for 3 samples. So now I'm going to show you the first 2 comparisons. That there are lots of things you can do here, but I'm going to skip here. I'm just going to go to the main plots first. You can always go back into data prep if you want to change anything. And so this section, there are lots of things you can do. So in the first plot, you have plot type. So there are some main options here that you can change the X and Y label. So the background of this data in the gray section is only 10% of the data. So I'm not putting all the dots just to increase the performance of that, because this is in your laptop, running on your laptop, and it requires lots of memory. So there are lots of dots here. And then to make this like interactive, imagine like when you hover this, it's just drawing all these variation here without writing a code. Usually, we were creating this by writing a code. And I was working with Melissa, and she was asking, what is this gene? And when you create a static plot, you don't know what that gene is. It's so hard to find it. So, and I created this. And when you hover, you know what it is. You know the p-adjusted value here, log 10 p-adjusted value, or log 2-fold change. So all the values are here. And when you click anything, it just keeps it here. So hover is not gonna work. When you click, it's gonna, whatever you click, it's gonna stay there. So other thing you can do with this, so you can choose a section if you want, like you just want to see this section, what they are. Like you can create a heat map here. And this heat map is not interactive. Again, for the performance issues, I just created like on and off button here, like interactive. When you click here in the heat map options, this create a heat map, and you can zoom in and zoom out here to see the gene names and stuff. This is one thing. You can create the heat maps like this, or you can change anything here. Like, you can do k-means clustering, your own data. If you want to cluster different genes, you can change the heat map colors to, I don't know, usually I use these colors, like, in the literature, usually red is upregulated, blue is downregulated. So these are you can use these. But again, this is two sample comparison, and we have this data options here. So you can change the normalization method with other TMM, Areli, upper quartile, whatever normalization you want to use, because we did DE now, we did the test, it's done. So while normalizing, while visualizing them, we normalize them, and they look beautiful like this after you normalize and scale. So this is the normalization method. And after you finish, like this is your table. In the table section, that you can see the genes and all the control and experiments here, P adjusted values, log2 fold changes and fold changes. If they are blue, it's in this case, it's downregulated, red upregulated. So you can download this file using this button and play somewhere else too. Like I told you, this wild statistics here. So you can sort this using wild statistics, and using this order, actually you can feed a GSA algorithm only using the gene names. But when it's ordered, it's good to go. But when you're doing this, don't use up and down only for if you want to run GSA. Just use all detected genes, because you want to give all the genes in this order. Okay, when you click here, so all the genes means all detected genes, that 14,000 genes after filtering. I have this here, and then if I sort this on this order, so you can either up and down regulated. It's not that important, but this order is important while running any ordered GSEA or go-term analysis. So you can download the data here, or you can search your data here or here. If you search it here, you can use that search. For example, if you search FGF genes in all detected, see, I changed this to all detected. So all FGF genes, only one of them looks like significantly up regulated. It's FGF21. So in our homework, actually, we did this. We know that this was upregulated. The other genes, FGF genes, are not upregulated using two samples. So, and there's another option here that you can put these two comparisons together into the same table. See, I'm just going to remove this FGF for now, just to show you. See, this comparison table is just get the union of two sample comparison versus three sample comparison and put them together. Sometimes you need to do Venn diagrams. You can use this this table. You can download this table, so you have these values. Fold change comparison 1 versus comparison 2. It's here. See, for example, when I have two samples, this is not up. This is not significant, but when I have three, it is. When you're increasing your power when you have more replicas. This is telling you that, these comparisons are telling you those things. So this is, for example, again, like it's on the border, so you can decide you want to get this or not. So this is not upregulated. It's significant, but this wasn't upregulated before, downregulated now. It is. So sorry, this was before downregulated, now it is not, when you have three replicas. So these kind of things might want you to use three replicas. So okay, so again, you can download these tables, you can do your search. Another cool thing that you could do So you can always come here and change the data set up and down again, because it was trying to draw all the compare all the genes in that comparison. So, okay, I have this, for example, there's one gene here. Let me zoom in there. It's not significant. Why? This is not significant. Even fold change is huge. It's because when you click, see, the variation is huge, like 300, 500. That's why when I use DE-seq2, it's so stringent. Like it's even taking into account this kind of variations and assess this as this is not, the p-value is like high, more than 0.01. And but sometimes, you know, you just don't want to be that strict. You can change the p-adjusted value here, or fold change according to your need. It's not God given. These things we use sometimes, you know, like 0.2, 0.5, whatever, but according to your need. For example, I was working on one protein, TDP43. It is tightly regulated gene. When you increase the fold, like, you know, when you upregulate that gene 1.2 times, mouse dies, you know, rat dies actually. And it's like, you know, 20% changes actually kills the rat. So that's why, you know, some genes, even the fold change is not that high, but it can be really significant, upregulated, but it's like very tight regulation there. Like it's just controlling in very tight, but can be significant. That's why sometimes you change this to 1.2, for example, and to see, are there other genes, like even the fold change is not high, but most of the genes are coming here. They look like significant, but then you need to increase the p-adjust, decrease the p-adjust value like crazy, just to see that are there any tight regulated genes that might be significant. So you can look at them, see. So it looks interesting here. So you can look like this too, 1.2, but this is very, very like tightly regulated genes that you can get that list and do your go-term analysis. You can trust this more because this is very low chance that they produce similar values. So see, they are like this, for example. Okay, so there are lots of things you can do. Again, you can do like your volcano plot. You can do your MA plot that you learn how to do. You can download them in different PNG, or you can change this with another like you can download as, there is an option here in the, where was it? This is a MA plot I'm looking for. Sometimes I forget things. Okay, here, download SVG. So sometimes you download this as a support vector graphic that will be easy for you to change in different programs. This is PNG and this is not vector. So you might need to do that. And the other thing that you could do, like for example, you can search your gene here, FGF21, and you're curious about where that gene is, FGF21 gene. So it's going to be green, or whatever gene you search, it's here, see, FGF21 gene. So if you don't see, there's this checkbox here. When you click it, you can put the name on it, like FGF21 is here. Sometimes I use this, especially if there are a couple of genes, I'm curious just to show them, hey, these are here, and then put into your supplementary. So these are things that you don't need to write code, you know, if you like these things. And the last thing you can do, like before you doing these things, there are also QC plots here that you can, this was like 2x2 comparison. If you want to go to the second comparison, there's this filter section, and you can change the comparison type in that section to choose the 3x3 comparisons. And when you do your PCA plots, don't use only up and down, just use your all detected genes, okay? Because this is going to use unbiased way, it's not going to use only up and down. Again, question? Are there questions? Okay. So it is going to use all these genes in this table, regardless of they are significant or not. You know, all these genes, regardless of they are significant or not. Oops. It's going to do the PCA plot. And if your plot is like this, by the way, you can play with your PCA plot here too. There are lots of options about the PCA plot. You can do the same thing you did in batch effect, like you can put the text, you can select the legend, color fields, and you can change the margins if you have lots of data. So you can make it bigger, smaller, according to your data. So, but they look good here, like controls clustered together, experiments clustered together, so it worked. And you can apply your differential expression analysis. This is like, you know, post-processing QC, so we call this like that. And if you just want to draw most varied genes, so this is going to do the same thing like PCA is doing. Actually, it's calculating the variance of the samples in this line, and it's just bringing most varied genes into the list. You can change top 500 most varied genes when the total minimum count is 10 in the row. So you can increase this value. So these are all interactive. Table is changing now. You're looking different part of your data by changing these parameters. And then what you can do, you can change the plot type. Now you can go to QC plots again. So I just choose this portion of the data, and you can change the plot type. For example, I'll show you second plot type, like all-to-all scatter plot or heat map. See, I didn't use the data coming from differential expression analysis, but this time my data is most varied genes, okay? And even by looking most varied genes, without using the DE information, data doesn't know that this is control, this is experiment when you apply this most varied data set. By just looking by eye, you can see two distributions here. It means that your data is beautiful. See, when you compare experiment versus control, it's just started separating. Like when you change this most varied genes, you can put like top 1,000 genes here. It's going to update these plots like immediately. You don't need to write code. See, I move like 1,000 genes, but if you want to go like further down in like more abundant genes, you just increase total minimum count. So you can actually browse your data yourself. That's why we call this DE browser. So these are like more abundant genes. You can look at the numbers. I just cut this off. And because my total minimum count is 1,000, so you can see like these are experiments, highly correlated, controls highly correlated, and you see two distributions. So by just like without doing any DE, even this tells you these genes, most of them are significantly different, but there are these things are not, you know, because there's some variation and it's not, then you need to remove this. That's why we do DE. But there's another plot you can do, like there are 1,000 genes. I'm going to make this 500 for now, just to finish it faster. And you can, yep.
Sorry to interrupt, but can you also show us how to make Venn diagrams with these?
I don't have Venn diagrams here. I didn't put it. Actually, we should. I didn't do that, but it's a good, you do Venn diagrams. How I do is like, I just download this list in one list and I download another list choosing, and there are like, or how many lists you want. And then there are like Venn diagram online, you know?
Yeah. I was just wondering if you could do with this as well.
I didn't put it in it, but maybe actually we can put it. Arthur is really good in Venn diagrams. Maybe he can put it in.
Yeah, so yes, I mean, whenever you guys believe that something should be added, just say so, because we keep developing the app. Exactly. So we do that all the time. Don't worry about it.
And we are using too, like, you know, Venn diagram. I wasn't using that much. That's why I didn't put, but I see some people are using a lot, especially actually in another program, Arthur did that. You know, he can show maybe to you, Kashi, later. So we can put that thing here too. It's not a big deal. I think most of the things that I showed you, you can play with this. We have a tutorial there too. in the bootcamp session, go over it yourself and play with your data if you have. Otherwise, play with this demo data. And again, like, you can learn a lot from your data without even writing a code by just playing with QC plots. I was going to show you this heat map. So this is the heat map. This section, because I'm using most varied genes, now there are like some genes here on this side, for example, they cluster together. The variance is huge here, and we want to remove them. So when you change the data set to up and down, there's also up regulated genes, there are down regulated, see, they're removed. So that's why we apply the algorithms. It looks beautiful now. And using, for example, up and down regulated genes, you can do your go-term analysis here that we have. For example, this was mouse data. I'm going to do enrich KEGG. I'm going to change this to mouse. And then when you do this, hopefully, because I changed this, I don't know, it's going to come, but it should come some. And you can change this p-adjust value for separation of up and down regulated genes. And there is also another p-adjust value here just to filter these found results, that because it's calculating another p-adjust value, then it measures like the genes in your gene set is enriched in any category, in any functional category or not. In this case, this data is enriched in like these kind of diseases. Beautiful. Coronavirus disease came. See, this is new. This wasn't here before. This is the database in the background. When you update the tool, they update the Go terms too. It's beautiful. And for example, if you want to see, okay, if it's important in COVID, I don't know if they have it here, but I'm going to try. First time I'm trying. Let's see. So you can get the DE genes, but also you can get the KEGG pathway here just to see where your genes are up-regulated and down-regulated in that pathway. See, this is that coronavirus disease pathway. First time I'm seeing this, interestingly. And so this is the legend here. Like you see, these are down, these are up-regulated genes. So we see, for example, this gene is down-regulated in this data. These genes are all down-regulated. And these two are up. So this is whole pathway. So you can download this. And there are like download button. See, there are download plots button. You can download them and play. And you can create different kind of things to disease analysis. You can do GACA here too. And or there's also dot plot here just to show like these are diseases. The size of the data is showing the number of genes in your data up or down regulated. These are together, both I use up and down regulated. You can just use up or down if you want to. And yeah, QC plots, Go plots are like this. The tables are showing these numbers in that category. Huntington disease, there are 887 genes. 96 of them are in my list, up or down regulated. Cool. So you can do many things with this. Not everything, by the way. Don't think that you can do like Venn diagram or, these lists that I am surprised it's getting better and better, but usually it's not complete. All these Go categories here. By the way, in Enrich Go, it's doing now. Go analysis, you can choose the chemical component or molecular function or biological process. So though you can change the ontology too. So in this case, I did it with Enrich Go with, this ontology, and where it is in the in your cell, so you can get these kind of lists too with Go analysis. I think I'm done. Maybe we can start with the rest. If you have any questions with this, please ask. And if you need new things, new functionalities, please let us know.
Alper, I guess we have one question from Lindsay.
Okay.
Asking, how did you access Discover tab from data prep?
How did I? From data prep, how I came here to main plots? So there is a button there in data prep. So go to main plots here. When you click this button, it just starts drawing everything else. Okay. Was this answer the question?
Oh, I believe when you switch to main plots, the tab on the left changes automatically as well.
No, but
So that's
at the beginning, we don't have these things. I don't draw them
I
until
see.
you press this button. Go main plots button. This button is important. Okay. Okay, Arthur, it's all yours.
So
Want to do it now or give it 5 minutes?
shall
break?
we have a break
Yeah.
or talk about some stuff and then do it?
Okay, let's do maybe 10 minutes break, huh?
Let's do a 10-minute break and come back.
Okay, perfect.
Okay.
And I can answer any other questions, or we can answer.
Can I ask a question?
Of course.
Actually, I missed the earlier part of this thing, so I don't know how you get that debrowser from web-based,
So
because I couldn't find in the biocore website.
no, you can either install this into your computer or you can go to biocore.umassmed.edu. So this is online.
Right, but
version.
but
This
I
is-
installed mine, and then version is a little bit different. Is it okay?
What's your version? Like one? Two?
Yeah, 1.18.3.
Most likely your R is old. If you upgrade your R, you can get a better version. Means we always add new things. We always fix some bugs. That's why I would prefer upgrading R and R Studio all the time with the latest version, and then you can upgrade your browser too. Because as you can see, yep, tell me.
Yeah, when I installed yesterday, definitely I got that message asking whether you're gonna upgrade. So I placed an or session. So I was expecting that it was upgraded already, updated.
But the problem most likely, let me kill this. So when you want to stop this, it just dies like that. Okay, when you stop it here, it kills it.
I don't see your screen.
Okay, sorry. So you see now, I guess. So, okay, so here you need to do session info like this, just to see what your R version is. See, my R version is 4.1. This is the latest version. See, 11 2021. So when you install this version, so your R, your browser version should be somewhere here.
Oh, let me see. Maybe. I think mine is R Studio, not R. Maybe that's the reason.
This is R Studio 2. Mine is R Studio 2, but you have R inside of R Studio. See, you can tell, you can look at this console on the left, and there's R, and you can tell the version of R here.
Okay,
so
let me see.
On this side, you can just write this session info here. Also, you
it says 4.0.5.
405. So see, it is old, but there is something you can do. It's maybe maybe harder, but there's always this. Like, you can come here to this. You can search the browser and come to this page, and there is a source package. You can install it from the source. Like, copy link address here. You can copy this link address, and then come to your here. For example, you can do this curl -o this. If you paste this will bring it to your machine, all this file, and then you can run this rcmd install browser. See, when you do this, this will install it to your R. It should work. See, when you do this, it is going to install. Because I didn't change much from 1.18.5, I didn't add a new packages or something, so it should work like this. So in the future, if you see you don't have one version of a software, you can always go find the software, find that zip, gzip, and you can manually install like this, rcmd install the browser, rather than using install. See, this is installing now.
Mhm. But
And
also, is it possible that I can try from the beginning, like delete all the program, then I just reinstall, then it could
no,
solve problem?
in your R version, it's using a previous R packages. So like, you know, it's always like this. So if you come here, there is like bioconductor version release 3.14. Most likely yours is using 3.12. Means that the old packages. So in time, we add a new release, like we are changing the code, fixing bugs. Everybody is doing that in bioconductor. So in your case, you need to either upgrade your bioconductor version, like bioc manager version, or you can like update your whole R version and then upgrade bioc manager version. So you can even, like, sometimes I was keeping multiple R versions in my computer, but now I'm just using the latest version. But you can even like do that, but it's more advanced. But it's up to you. Like, you know, it's easy to install new R version like this. You know, you go R, download. So it's going to immediately replace it. So you're going to go to R projects. Either you are using Windows or Mac. You can go to, for example, Windows, and you can go to download R and choose one of the mirrors. And then you can download like macOS, Windows, or Linux. If you click, I always come here and I click this. And then when I click this, I come to this R 4.1. When you install this package, it's gonna upgrade your R immediately. And then
I see.
and then you need to restart your R Studio. Here, there's an option here. After you install that, there is this session, restart R. So when you restart R, it will start using the new R version in your computer, and try to install something or like the browser again, and then it will upgrade all the rest of the dependencies. There are 200 dependencies my like the browser is using. Yeah, here, see, there are 209 dependencies, so it will upgrade all of them to the new version.
I see.
Hopefully. So it
Oh
works.
yeah, I will try.
Yeah.
Oh yeah, thank you so much.
You're welcome. But these are the things, like when you start using R, you're gonna do these things a lot, like, you know, use a new version, upgrading your system. But again, if you want to reproduce the old results, use Dolphin Next, because we always keep the old versions of anything in our containers, that you can still reproduce your R results that you created a couple of years ago. So make sure to the, like, for example, I was working yesterday. See, I create this R script and I put it inside, and this creates a heat map, for example, and it's using some versions of ggplot, like all these things, but everything is in the container. Like 2 years later, when the new versions come up, when the functions change, this will still work. But it's not going to use the new versions. It's going to use exactly the same version I created today. Okay, this is very important, because you're going to see in the future, your code will not work again in the future, because these functions will change. Like, people are changing their code all the time. Okay.
Yeah.
9:55. I think, Arthur, you can start. I'm just
Okay.
gonna make you.
So just to remind you guys again, if any of you got kicked out from the Slack channel, I believe Onur has sent you invitations again. But if you're still having problem connecting Slack, I am going to try sharing the links that I shared in Slack as well as Zoom as well. Can you guys access Slack now? Did you get the invitations, those who got kicked out? Okay, you can accept it and get back to the Slack channel then, Sandhya. Okay, I guess, okay. All right. So I guess I'm gonna start. We can fix that along the way. Let's see. Okay. Thank you, guys. So we're gonna move on with single cell analysis now. I believe this is the second time that we are actually teaching single cell data analysis and single cell pre-processing here in the bootcamp. Last time we only did it for one lecture, I believe, Alper, was that the case? And we kind of believe that it's a little bit more extensive. We have to touch base with more machine learning algorithms, some more statistical methodology to actually explain what why we are using single cell analysis the way we do. But this time we actually added a couple of more exercises and hands-on practices for sequencing your own data with Cell Ranger and 10x genomics tools. So we're gonna just do all that. So basically, what we're expecting for you guys to be able to do after these lectures is that to understand the basics of single cell data analysis, we're just gonna work on really simple examples to cover the fundamentals of stuff like clustering, which is really important to discover different cell types within a the cell population of a sample. After single cell sequencing, we're gonna see how we can pre-process and quantify single cell RNA-seq sequencing reads, and we will employ perhaps the most famous single cell RNA data analysis tool in R, and we will start doing UMI distribution plots. We're going to see how to filter, normalize data, similar to RNA-seq, but it's slightly different. And we will see how clustering is actually used, which is really essential to single cell analysis. And we will see, not today, but on Thursday, when we move on with the more advanced stuff in R, how to annotate these clusters and how to discover cell types existing in our sample. So basically, what I'm asking you guys is that if anytime you believe that I'm going really fast, and anytime if something is not clear, you can just stop me. We have a lot of time, so I can just stop and answer your questions, and we can just cover whatever was not clear. So question, did anybody try installing the package called Surat in their R Studio? Anybody who didn't install, had a problem with it? No? Is everyone good?
I tried installing, but it told me that it's not available in my R version, and I cannot download an earlier or a more recent version of R because my computer is not compatible with it. So I'll have to try on a different computer. Yeah.
So we had some instructions to how to install RStudio in R in session 6. So this version you can't install, 405.
Sorry, you said I can or I cannot?
Can you install this? No.
I don't think I can, no. I have 3.3.3, which is, I think, the latest that I can install.
I see.
Yeah. I'll try on a different computer.
Okay. So anybody else? You're good? Okay. So as we are going through the course, you can actually try installing it, and if there's any problem, Alper and Onur may help you as well, and I can help you too. So along the way, it may ask you questions like, if you want to install the package from the source without compilation, you can say no to that. If there are old packages that Surat is depending on and you need to update, you can just say yes to all. It may take some time, but Surat is a really extensive package that use a lot of different packages in the same time. So you really may need to update all of that. But since Surat is a really popular package, I believe that it's going to work if your R version is recent. So what are we going to do? Basically, we will, cover what single cell RNA sequencing is a little bit. And we're gonna talk about why we actually need to use single cell, for what purposes. It's much more advantageous to RNAs, regular RNA-seq, which we call bulk RNA-seq, and we're gonna talk about the differences between them. And we will see how we can do scRNA analysis and sequencing and quantification alignment with Dolphin Next. So the reason we especially focus Dolphin Next over here is that Dolphin Next is actually allowing you a great interface for you to easily run pre-processing pipelines, and you actually don't really need to use the terminal, even though in some cases you may, if you are doing something really new, but you can always develop your own pipeline Dolphin Next. But we're going to especially show today to use a simple pre-processing pipelines that processing 10x genomics reads, and how we can do that in Dolphin Next. And we're going to spend some time on it. So then we're going to talk about how RNA-seq data structures are structured in both R, and how the results are actually quantified. So that's going to be really important, because there are specific features of single cell RNA where you have multiple, thousands of expression profiles coming from multiple numerous cells. And in order to store that kind of data that has a lot of zero reads, we will going to incorporate different kinds of storing methodologies, and we're going to just kind of cover all that. Then we're going to start slowly start analyzing data in R. We are not going to go into dimensionality reduction, clustering, or mark analysis today. We're going to do that on Thursday. And today, what we're going to focus on will be how we can import data to R, single cell data to R, how to do quality control, how to filter out some cells that you believe that have less quality than the majority of the other cells that you captured, and how we can normalize them. We're going to stop. And along the way, we're going to have some homework for you too, that you have to complete until Thursday. But you'll be able to follow regardless. So basically, everything that we're going to do today, we kind of saw how to do them in some way, right? We know how to use the cluster, how to manage our data in the cluster environment. We know how to do RNA-seq analysis in some way, how to use R packages, because RPair covered the browser, some other libraries like DESeq2 that has a lot of built-in functions. By the way, when I say a built-in function, it means that a function that was written by somebody else and put it in an R package, so that everybody who imports that package, like Surat, can actually use those functions without actually writing too much code. In a way, you're writing some code, but it's really minimal. So this is how we actually share scientific methods. Whenever we have a new method to analyze some advanced problem, we build packages for you guys so that you can easily use it and reproduce the data. The browser is one of those. It's actually a package that allows you to execute an interactive interface where you can do the analysis yourself without writing code yourself. And it's using other packages too. So R is a large community and large environment of packages that all packages are depending on each other, and there's a fine control on whether a package is old. You can always go back and update, because whenever you download a new package, it actually asking you if you want to update all other dependencies. So everything is smooth. Just install the library and you're going to get everything that you need at the time. But again, I mean, installing R packages are troublesome. Sometimes you may come across the problems. If there's anything like that, you can contact us again. And if you're downloading the R package Surat now, if you have any problems, you can just go ahead and ask. Okay, nice. Okay, so what is bulk RNA-seq data? Basically, what Alper was talking about for the last two sessions, the Thursday and today, actually is that what we call a bulk RNA-seq, because compared to single cell RNA-seq, you actually have a bulk population of cells in each biological replicate. I mean, this is really nice, right? Because at the end of the day, in order to understand certain mechanisms in biological experiments, you have some control group where you have multiple biological replicates. Then you either apply some kind of an experiment, you treat those cells or you treat those biological samples with some perturbagens, or let's say that you have the same number of diseases, same number of people that have some kind of disease. You can understand whether which genes are up or down when the disease is active in a patient but not in another patient, or the gene is actually upregulated and downregulated when you treated the control sample with perturbagens or other stuff. But the interesting thing is over here is that it's nice to see what genes are going up and down, so you can come up with drugs. You can have some understanding of the signaling pathway that it works when you knock out or upregulate some gene in the biological replicates. What's really happening in some cases where you have an heterogeneous population in your sample, you have T cells, you have natural killer cells. You may have, we're going to give an example from skin data. When you have keratinocytes, you have perhaps macrophages. So each of those are actually reacting in a different way when you have a disease or when you're treating these samples. So it is actually some kind of a signaling across cells, that cells are, making genes. They are, coming up with, chemokines that signaling other cells to recruit some other cells to come into the disease area. And it's all about, knowing what kind of genes are going up and down whenever there's a disease or some kind of a treatment. So basically, what we are trying to do with single cell is to understand that, right? Not just to see how the genes are changing in totality in the biological replicate, but we're trying to understand how different cell populations are reacting to different conditions, right? But what we are going to find out about in these lectures that how we can actually find those different cell populations to start analyzing what changes when you have different conditions in your samples. By the way, in these lectures, we are not going to cover the analysis across different conditions, even though that's what we've been talking about, because it's a rigorous process to find out about different cell populations in your data. Sometimes you may actually have similar biological replicates in your data, but you're trying to find new cell types that may have different phenotypes. So that might be the case, but since it's a little bit more advanced, we are not going to cover that, but we're going to cover until we have been able to find out different cell types and annotate them. So why this is important? Let me give you a really, really good example. So beside Kashi and Nazgul, who knows about skin, or do we have any dermatologists in the course now? No one? Okay. So me, Alper, and Onur, we are bioinformatics core, but we work closely with Manuel Garber Lab here in program of bioinformatics that who also collaborate with Harris Lab, John Harris Lab in Department of Dermatology. And John Harris's specialty is vitiligo, a disease where you have certain areas in your skin losing its color because your T cells in your immune system are turning against your own cells, specifically melanocytes that are making melanin, which gives your skin your color, and they are killing them. So basically, with this problem, you have white patches in some parts of your body that gives you these kind of weird spots in your skin that are white. You may have really large patches, but you may have small ones as well. So it's really interesting to find out that why T cells and how T cells or what kind of T cells are actually killing melanocyte, which is really, really important to come up with new treatments to understand which kind of signaling pathways that we have to block in order to block these disease and eventually cure it, not just to treat it. So just to understand why single cell could be really, really important, we have to actually just cover the basics of a disease like this. So what's really actually happening is that CD8 T cells, they're coming to your skin, they recognize melanocytes, right? And they start making interferon gamma. And when they make interferon gamma, they start instructing keratinocytes to make CXCL10 and CXCR3. And this actually triggers more CD8 T cells to be recruited into the skin from the blood and eventually killing more melanocytes, right? So the one interesting idea is that if there is some kind of a recruitment signaling pathway that is happening in interferon gamma that is specifically expressed by CD8s, then maybe this signaling pathway should be blocked, right? And that is what was happening. And that is what people in Harris Lab, they actually did it. But of course, even though they found out about the signaling pathway in the major populations, what happened was that after the pathway was blocked and the treatment was working, when you stop the treatment, the disease relapses. So you start losing melanocytes in your skin again. And what they found out about was that it's a subtype of CD8s, what we call resident memory T cells, whose CD8s that stay in the disease site. That's why we call them resident memory cells. And they stay over there. And when you have more melanocytes, they recognize them again. Then this pathway occurs again. So basically, you shouldn't be stopping the treatment. But again, understanding these pathways, coming up with new ones, is only possible that you can actually figure out which subtype of cells that is that we have to target in order to treat the disease, and which gene exactly is upregulated and downregulated in which one of these subtypes are really, really important in order to find out about the disease progression. So single cell gives you another layer of understanding cellular mechanisms, not just to tell you that, what gene is up or down, but it actually tells you in which population it might be up or down in different conditions. And it may also allow you to, find about new T cells, especially when we had another case study with our collaborators back in Harvard. We were working on another skin disease called Stevens-Johnson disease, where we started, finding new T cell subtypes, and we started understanding that there are actually around 90 different T cell subtypes recorded in the literature, and T cell methodology is quite vibrant. You may come across with a different T cell with a different phenotype anytime. So it's in this case, single cell analysis is really important to find out about these subtypes and understand its phenotypic profile. So how do we do this? So basically, in the case of vitiligo, there are these special, tools that it sucks your upper layer of the skin, the epidermis, and you basically can extract these cells from the epidermis, and you can use special single cell RNA-seq methods to isolate the cells, and we're going to talk about how that's actually possible, and how you can actually come up with expression profiles of each sample. So this is really interesting. So you see, this is each points in this plot are actually a cell, and you have a lot of them. Specifically in the sample, in this data, there are about 33,000 cells where we have access to each of their expression profile. And we can definitely see different subtypes of cells. We can we can just isolate the T cells, which are lymphocytes over here. We can see which of those have more interferon gamma or not. So, but in this case, there are only 10 patients over here. So this actually shows the power of single cell analysis. With just 10 sample, you can generate data with numerous expression profiles that gives you a lot of room to do detailed analysis. So dimensionality reduction is really important, especially in single cell data. But this kind of dimensionality reduction is actually unique to single cell data as well. So that's why I believe that single cell data is a little bit more mathematically complex that brought out the potential of advanced machine learning algorithms, and we're going to talk each of them briefly, and we will see how we can easily execute these algorithms without knowing too much any of them. So, dimensionality reduction is a thing for all kind of sequencing analysis. And as you can see, we actually did that in bulk as well, right? So basically what we've been doing was we had a control group and we have a treatment group. And once we have some differentially expressed genes, we can actually reduce the dimensionality of those expressed genes and see if there's a fine separation between the groups. But what's really important over here is that you already know the groups. You're just validating that if there's a fine separation, or you may actually see that one sample's expression profile is slightly, away from the other two or other samples in the same group, which may indicate that its expression profile is slightly different, right? So, but the funny thing is that we only have one expression profile for each sample over here. So there's not really much room to identify heterogeneous, patterns and behaviors in the expressional programs. But with single cell analysis, you can actually generate numerous expression profiles from each sample that will give you more room to detect. But the problem will be that we're going to talk about, and I'm going to repeat it again, is that initially we won't know which cell is actually within each group. It's not like we know as soon as we have the expression profile if these are macrophages. This is short for macrophages, by the way, or some cells are actually melanocytes, which here is the short for melanocytes, right? So basically the fundamentals of single cell analysis is that you're trying to understand what kind of different expression profiles and groups and clusters in the data, and then you look up their genes and which of those genes are high in which of those clusters. And we're going to start telling that, okay, these are melanocytes, these are definitely T cells because they have CD3, and these are definitely melanocytes because they have TIR or PML, those genes that are really indicative of cell types. So basically, yes, the idea is to isolate RNA from each of those cells, tagging them in some way so that we know that those RNA fragments are from each individual cell, or what we believe from each individual cell. We're going to talk about why an RNA may not be or be from a cell, even though we do these experiments, and even though the objective of the experiment is to isolate cells. But we're going to talk about some technical difficulties, which are the essential of single cell and RNA sequencing. And understanding the technology is really, really important to know how to filter and do quality control of it, because if you don't know how those cells are isolated and what problems may occur during cell isolation, you don't really know how to interpret low number of reads that you get from one barcode. We're going to talk about what barcodes are, or so forth. So basically, to summarize, what we have is that you have some tissue where you dissociate the cells first, but it's may or may not be a mixture or a cocktail of cells that you may need to know which type of cell types are. You may really only need to see if in a treatment case some genes are up or down. Then you can just toss them into a library, get the RNA, amplify it, and see how many unique molecules that are associated to genes after their alignment you have for each biological replica, right? But if you really want to know if some certain cell types are different from each other, and how many of molecules from genes do they belong to which cells, maybe those genes are only active in T cells, that you want to know that. We were actually basically talking about that with Alper in one of our last meetings, and the question was coming up, okay, is this in T cells or not? So that's basically the objective of single cell analysis, to know which genes actually belong to which cell types, right? So you don't really have multiple expression profiles. I mean, you don't have just one expression profile for each sample. You have multiple or numerous or thousands of expression profiles from one sample that you start doing clustering and understand cell types, right? Talked about all of those. So basically, the understanding is that you just don't have one expression profile, you have many. So you can actually observe the distribution of the gene as well. So you may actually see the gene up in bulk, or let's say that you can sort out T cells. Basically, it's not like we need RNA-seq analysis to isolate T cells, right? We can just use FUX sorting to sort for CD3 and just isolate CD3s from all other cells in the specimen, and still do some kind of the analysis for treatment versus control. But basically, we don't have a distribution. Sometimes, even in that case, you may have a bulk aggregate count of each gene, but basically, you may have only one or two cells having that gene up in randomly. It may actually be possible, and the most of other cells in that subpopulation may actually have that gene 0, or you wouldn't be able to find any reads on that, right? So what single cell is actually doing, other than spotting different cell types and different populations and allow you to do the analysis on those populations, actually telling you the landscape of the expression of a gene in certain subtypes. So you basically may have some mean expression profile, but some of those cells will have fewer number of expression in that gene, and that's what we want to know. We want to know how many of those cells in one subgroup or subpopulation have high number of non-zero counts of that gene, and that's basically what we are trying to understand in single cell analysis. So let's cover the process a little bit. You have a tissue, you have an organism, or you may have a cell culture, you dissociate cells from it. Then what we are trying to achieve in some way or the other, and this is all the reason, the objective of single cell technologies, is to create some kind of single cell isolation. The reason for that is once we extract the RNA, we are trying to see that all the RNA within that isolated space actually have RNA from the same cell, or potentially from the same cell. We're going to see some examples of it may not be the case. There may be two cells within the same isolation. It is possible, because it's a really, really hard problem in practice. And but there are kind of methods that you don't really isolate, but you're trying to make sure that the RNA from different single cells are tagged in such a way that different combinations of barcodes and the molecules that we tag with RNA that indicates the cell of origin is actually unique to those RNAs. So there are different kind of methods. We're gonna talk about all of those. So basically, you isolate the cells and you tag or barcode the RNA fragments from the cells, and then you start making your library, right? You amplify the cDNA in different ways, and you come up with your library, and we move on with sequencing those reads and try to look for those tags that are specific to those RNA from certain cells or certain unique fragments, and we start sequencing. But what we really want to do in these lectures today specifically is to see how we can actually start with the sequencing read that we capture from the sequencing machine, which tools to use to align and quantify these reads and understand how many of unique molecules from some gene actually belongs to one cell or to another cell, and how we can come up with these single cell expression profiles where we have rows as genes and columns as different cells or supposedly cells, and start doing some analysis on it and incorporate machine learning methods to dimensionally reduce the Gini mathematical space and start identifying the cell types. I actually like this figure from this particular paper a lot. It's a simple review to the developments of a single cell RNA-seq in starting like in 2009, and it briefly explains the differences between different technologies and how they actually started exponentially increasing the high throughput, the number of cells that you can capture in a single experiment or multiple experiments, and how we have these unique commercial-based tools to easily capture different kind of expression profiles from cells and analyze them. There are some other tools as well. We're going to talk about it, but you may actually recognize 10x Genomics here. And basically, we will be using an example data from 10x Genomics website today to analyze our single cell expression profiles, but be mindful that you can actually use any kind of method to generate your single cell expression profiles. But again, the quality control and the performance and the accuracy of the analysis will actually be dependent on the sequencing technology that you're using. So I urge you to take a look at this paper and read in detail. So there's a link over here. You can just click on it and find more about it. So basically, in 2009, we were only able to come up with the expression profile of a single cell, right? Because the single cell isolation was not as advanced as it was now at the time. And basically, up until 2015, you were either efficiently or inefficiently needed to isolate the cells in different independent wells of a plate, whether it's a large plate or not, and it's actually time consuming. So starting from 2009, it was much more simpler. Then some methods came out that made this isolation through separating into wells process in a much speedy way, using flux sorting and other kind of some fluidic technologies, but it was never as efficient as we were using today, which was initiated by methods that usually have this abbreviation DROP in it, which is referring to the type of single cell sequencing technology, where we don't actually have to separate cells into individual wells, but we can actually have these system of nanotubes a system of microfluidic technology that feeds cells in such a way that we are trying to make sure each cell will be isolated in a single drop of oil, where we will be able to, put some reagents along with the cells in the same drop, to such that those reagents have the necessary nucleotide sequences as tags. And when we align them, we'll be able to align those tags as well and know which transcript each from each cell. Of course, it has some difficulties. Sometimes you may have multiple cells in a single droplet, but we're going to talk about all of those. But first, we're going to cover that. There are multiple methods that use the method DropSeq, which is short for droplet-based single-cell RNA sequencing, which was initiated in 2015 in a group in Broad, I believe. Or the author of the first paper could be in Broad and have a little lab over there. I don't quite remember now, but it's been further its efficiency was further increased later by 10x Genomics, and we're just going to talk about that too. So what's happening basically? Let's see DropSeq. But by the way, who did single cell analysis over here? Who actually had a single cell experiment before, isolated some cells, and totally knows what I'm talking about? Can I have a show of hands to see who did that? Okay, Alisa did. I know Kashi and Nazgul did, but they're shy for some reason. You could just say so. So I guess Kashi and Nazgul were using another technology, but we are not going to talk about that today, 10x. But I'm not sure Kashi and Nazgul actually ever did 10x. Did you guys did ever?
Yeah, I did 10x with hashtagging.
Oh, you did that, right? With SiteSeq. Yeah, it's a slightly different technology, but it's using the same protocol in some way. But yes, I see. Okay.
Just a question before we go forward. Do you also have the hashtagging in the DolphinX for analysis later, or no? That's something that will be added, or?
So I mean, I can talk about it that later, but yes, we have a potential pipeline, but it's still in development. We're gonna make it public really soon.
Oh, that's great.
Yeah, because a lot of people are actually asking that. It's a good thing that you remind me of that, Kashi, but we can maybe talk about it later too. But we are not going to do any hashtagging, or we are not going to prepare essays from additional essays. No, we're just going to use RNA.
Great.
At this point.
Thanks.
Okay, so let's try to understand. So basically, we have a complex tissue, we isolate the cells, and we're trying to come up with such a way that each cell is suspended in a unique environment, which will be a kind of like an oil that is we refer to as a drop, and we will be putting a particle called what we call a bead in it, and these beads will have nucleotide sequences unique to each bead, and it will have some number of unique molecular identifiers to identify each unique transcript covered from each cell, which is suspended in the oil, right? And this is how we actually identify transcripts that are originated from, supposedly some cells, right? And if you take a look at it, so when you lyse the cells, it is expected that the RNA will be dispersed around the oil, but it's not going to go outside, and it's going to attach to those unique nucleotide sequences on the bead. So, I mean, there are in fact millions of them, but just to cover, each bead has the same cell barcode, the same exact cell barcode in it, so that the assumption is that there will be one bead in a drop of oil, there will be one cell. So each of these RNA fragments and transcripts will be attached to these barcodes and UMIs. The barcode will be the same, and that's going to be the nucleotide sequence that allows us to identify that particular RNA is from that cell, right? But since and I believe that Alper actually thinks that this is really, really important to talk about, is that the coverage in single cell analysis is really, really low, and amplifying the RNA covered from each single cell is really important. So UMIs, unique molecular identifiers, are essential to most single cell experiments where you have unique UMI for each cell barcode in the bead, so that once the transcript is attached to it, and once we amplify the cDNA, we can actually know how many of the copies that we have from each unique RNA, so that after resequencing, we can collapse it back and count the number of unique UMIs in each cell suspension, just to say that how many of genes on RNA belong to those cells, but not just the copies, but the unique molecule and the gene itself. So basically, there are many number of cell barcodes that you can generate, and each bead has a unique one of them. And each bead is attached to numerous number of UMIs to capture unique molecules in cells. So
just a question I may have forgotten, but is the UMI sequence the same for each bead, or each bead has different sequences of UMIs?
For each bead, there are different sequences of UMIs. Right, because if you had two same UMIs, you may have actually captured different RNA with the same UMI, and when it was collapsed, it would be regarded as a single nucleotide sequence, and that's what we don't want. So basically, each bead has the same cell barcode but has different UMIs attached to them. So the UMIs is identifying unique transcripts, and cell barcodes are determining unique RNA from one cell of origin. Did that answer the question, Kash?
Yes, thank you. Yeah, I was just a bit trying to remember my.
I see. So basically, I mean, I'm not a biologist, by the way, so I am actually an original statistician. So if any of you guys actually catch something that I actually said wrong over here, but we're just covering the technologies over here. But just step up and you can just talk to me about this. So you can read more about the dropseq in this publication, which was in 2015, and learn more about the technology if you don't know anything about it. But one problem with dropseq was that it is really hard to capture one bead and one cell in one drop of oil. So basically, this is what kind of a microfluidic technology that we're talking about. You have beads flowing in from one place, and you have regions and cells flowing from the other side, and the oil actually comes from here. And just the assumption is that we have suspended one cell and one bead inside one oil to capture RNA from that cell when we lyse the cells in it. But the interesting thing is that, in a way, the cells, the beads, and the oil is randomly flowing in. So it is highly likely that you're going to have empty droplets. You're going to have a droplet with only one bead inside, on just a single cell inside, or you may have actually two cells in it, depending on the speed that the cells are being fitted into the microfluidic process. But basically, the efficiency of the method is based on the efficiency of how the speed of feeding in the beads, the cells, the oil is being optimized. So when dropseq came out, this efficiency was really low, only around maybe 5% of droplets only had one particle with one bead and one cell inside of it. And a big portion of the droplets were actually empty. You can, practically solve this problem with just increasing the speed in a way, but that actually will increase the probability that you have multiple beads or multiple cells in the same bead as well. So it's a really delicate process. There are some technologies that you can drive yourself using the dropseq, or there's another technology called indrop, but you may need to actually control this process as the cells are being suspended in oil along with the particles. But of course, we're going to talk about an alternative technology where you don't really have to do it. And they could be the most efficient technique around. So 10x Genomics was the company that actually made these processes more efficient. Their microfluidic technology is really similar to DropSeq. And the interesting thing is that we are doing the same thing, but there is this machine where you are feeding in the cells, the reagents, and the oil from different parts of the microfluidic nanotubes. And you just put it in, push the button, and the cells are tagged and suspended. And the RNA was isolated where the UMIs and the barcodes attached to unique RNAs. It's I must say, it works like magic. You just push it and wait for 20 minutes, go get a coffee, and it will do everything for you. And once you come back, you can move on with the library prep as instructed by the instructions in the 10x Genomics. So you can actually go and take a look at these products of the company. I believe that everybody heard of 10x Genomics here, right? Is there anyone who didn't know about 10x Genomics before? You can just raise your hand. Oh, Samuel, okay, nice. I mean, 10x could be the most popular one. So out of, I mean, 100 single cell data set that you can see in public repositories, probably, I don't know, probably 95 of them are actually from 10x. So let's move on a little bit faster, actually. So basically, the technique is really simple. You have barcodes attached to the bead, and the barcode is specific to each bead. But for on each barcode in the cell bead, in the bead, you have different UMIs that identify, you know, unique molecular reads before we amplify the transcripts. So after this, and when you prepare the library and put it on the sequencing machine, what you're gonna get is that you're gonna have some reads where you have the barcode first, which will be of some number of base pairs. Then you're gonna have the UMI later, and we're gonna have the actual cDNA transcript. So cDNA here. So basically, after we amplify, we're expecting that some reads actually share some UMIs, which may indicate that these are actually from the same transcript that we're going to collapse. And it's not like we're going to, you know, count the reads, but we're counting unique UMIs. So that's why usually when people referring to single cell data, they're referring to as UMI table. And that is the reason we're actually not counting reads, we're counting UMIs associated to the transcripts captured in the cells. And looking for that, you know, we'll be able to recognize each barcode, and this barcode will probably belong to cell 1. This will be cell 2, and this will be cell 2 and cell 3. And some of these will be sharing some UMIs, of course, and that means that we only have some actin B UMI from just 2 reads of these, just one of them. So basically what we are looking into in single cell RNA-seq processing pipelines is that we have the library, then we align them, we capture the barcodes, the UMIs, and the cDNA, then we establish what we call a UMI table over here. So this actually looks really, really similar to the gene expression profile tables that we have. Gene expression profile tables that we have in bulk RNA-seq. So let me go to the beginning. See, so this is an expression profile table, right? So we have some counts from some genes, but it's actually an aggregated count of these biological replica because we have many, many cells, and we actually captured an aggregated number of this RNA from associated to this gene from each biological replicate. But now we don't have biological replicates in these UMI table, but we have barcodes, right? And the interesting thing is that even though it's written cell over here, what we're gonna see is that this is actually a barcode. Basically, some of these counts could be really, really small in total, which means that the number of RNA that you found in the expression profile is low, which will be indicating that those beads were actually attached to some random cell-free RNA that didn't actually belong to any cell. And that's actually not a cell. And we shouldn't be incorporating that to the analysis, and we're just going to remove them. So basically, one of the statistics that we're going to use will be the total number of UMIs, but the importance is that in the beginning, what you're going to have in columns are not actually cells, even though we call them cells. They are probable cells that are actually barcodes, and it's our job to understand which of those expression profiles may actually belong to empty droplets. And we're going to move on with the analysis. So before moving on to the samples that we have, I'm going to ask, let's talk about this. So we're going to use this data that you can find in, I forgot to add the link, I believe. So we're going to use the sample. So it has around 1,000 quality reads, and the cell ranger, the pipeline to process the sequencing read that captured by 10x will be doing some initial filtering. So we are expecting that some number of thousand cells will be in this one sample of healthy blood donor, and we'll be working with peripheral blood mononuclear cells, where we have some T cells in it, some NK cells in it, monocyte and platelets, and we'll try to understand how we can cluster these expression profiles. Let me see. What is the proper number? It's actually, there is no proper number actually. It's based on your sample. It's, if you're talking about data analysis itself, I would say if you have more than 50 and 100 cells, it's really okay. It's nice after filtering and quality control. But it's based on how much it's based on the content of your sample, basically. Okay, thank you. Any questions, by the way? Am I going fast? I might be actually going slow, by the way, but we'll see. Just move on a little bit more, then we're gonna have a small break. So now the question for you guys: have everyone a project space? Let me actually show you this in great detail. So we covered this practice basically really fast before, but normally we ask you guys to deposit your RNA-seq lecture materials in your home folder, which would be something like this, home after and your username. But this space, as Alper told you about, like in the beginning of the bootcamp, is only off. Let me just maybe pull it off really quick. Is of only 50 gigabytes, right? So single cell analysis, especially, and the case study that we have today is actually demanding a little bit more than 50 gigabytes. So is the case with the DolphinX pipeline. So it is better to move to a space that you have more freedom to use more gigabytes and hundreds of gigabytes. So your project space should have a path starting with project, then the name of your group. In this case, I belong to Manual Garber Lab, and the directory that is specific to Garber Lab is UMW _ Garber Lab, but the name of your project space which will be different. And as we talked about before, the minimum amount is 2 terabytes and it's free. So if you have talked about it with the IT HPCC admins, they probably have given you some space that has at least 2 terabytes. Did I miss anything, Alper, by the way? So basically what we are usually doing is that in our project spaces, are you guys hearing me, by the way?
Yeah, I have a quick question about this. So I tried this and it looks like I don't have permission to, first of all, I can't find the lab one. I found like Timofey Kowalik, which is my PI, but there is no Kowalik lab one. So I don't know if it exists. And I tried the Timothy Kowalik one, but I'm guessing that's the private account of my PI, and it's telling me permission denied.
I see. So in that case, you can contact HPCC admins and ask them to give you permission. So basically,
but is that my PI's account or is that my lab's account?
so basically your PI is asking for that space, but then anybody that PI is allowing to have a permission to access files in that folder can actually have it, but you have to do it through your PI.
So the PI has to first ask for a space, or does the space already exist for my lab?
I guess you have to ask first.
Okay,
Yeah.
thanks.
Yeah. But then again, your PI can contact the HPC admin to give you permission to update files. So what we usually do is that, by the way, as we just talked about, if you don't really know which folder actually belongs to your PI, you can just simply go to project. It's really slow today. And do a simple ls to actually find the exact name of the folder. Here, I'm just going to move on to my folder and start doing analysis. So here we usually have our own folders as well. So you can just go ahead and make a folder for yourself as well using make dear Manukyan or something like that, which is my name and surname. You can come up with your name as well. And I'm going to go into that, and it's my space now, which is isolated from all other spaces belong to the same group, right? So basically what we asked for you was that you have to copy the entire folder of scRNA-seq from this path and just put it on under your username, under your group name path, create a bootcamp folder under that, and just copy those files over there. Did anybody try and couldn't do it?
Yeah, I had a little bit of an issue trying to connect to my lab project workspace. That might be because I was associated with a different lab prior to this, so I think I might need to reach out to the computing cluster. But when I checked for my lab's project workspace with the ls function, I didn't see it.
Okay. All right. So okay. So it's a permission issue. I believe you're gonna have to talk with the HPCC admins with your PI at this moment. Okay.
Can you please repeat, how do I see my project space? I can do the ls and it shows I'm in Michael Francis lab and it shows me UMW Michael Francis. But
UMW what?
Michael Francis.
Michael.
Yeah, Michael Francis.
Francis.
Yeah.
I don't see. Oh, this is not Michael. Hold on.
CHAEL.
Yeah, Kasturi and I actually share the same PI. This is what I was talking about.
I don't see any Michael Francis here.
Yeah, we have. But if I can share my screen, I can show you that it's at least my works come under UMW Michael Francis.
You don't see the Michael Francis, directory, right? I don't see it over here.
Yeah, I do not see the like
probably you guys have to talk to the GHPCC admins to open your space.
Okay.
Yeah, you can just talk to your talk to your PI and just ask him to.
Actually, I remember I asked him, and when we signed up for this class, our PI has given us a permission, and then we were only able to have this Dolphin Next account and everything. Is this something different?
I am not sure what's going on actually.
Okay.
Could it be somewhere else? There's a nearline as well. Maybe it's there. I don't know. You said Michael.
Francis, F-R-A.
I don't see it over here.
Hmm.
I don't see it. Either it has a different name. Maybe we can. Francis. No, I don't see anything with F or Michael that is here. Yeah, we can talk about it later if you like. So Alper, let's if you like, let's give a 10-minute break. And we can come back on 11 past 5 and just keep moving forward with the analysis. Is that okay with everyone?
Yeah.
Okay.
Yeah, let's
Okay,
do
so
that.
let's come back in 10 minutes then.
Okay, cool.
Nazgul, you can't accept the Slack invitation?
I'm having trouble. I used the code that they sent me, and then it asks me if I want to go in through pick a channel to go in through, which I have UVM Tribal, or to join a workspace, but the workspace for the invite is not listed.
Is this, are you talking about the project space, Anna? Anna?
Oh, no, I'm sorry. I'm talking about Slack.
Oh, I see. Onur, are you there?
I sent him a message.
Okay. Hmm.
I'm sorry, I thought you were talking to me.
No, it's okay. So, it's fine. I mean, I was talking to both of you. So I don't know what happened over there, but
Yeah.
it appears Onur actually fixed some of the channel memberships, but I don't know what's happening.
Is the UVM Tribal something completely separate? Like, did I mess up by going in through that? Or-
Hi guys. So I sent an email to Heidi to fix this issue, but she didn't turn me back. And
Right.
I can do only send the invitation. I don't know the real cause of the issue.
Okay. My only concern is, like, I'd like to go back and watch the classes, and I know they were posted there because I was on for the first few days. Is there somewhere else that the classes will be posted that I'll be able to access, or only on Slack?
I'll put them onto GitHub. So
Okay, that would be great. Thank you so
I'll
much.
put every week there so you can watch them.
Okay, thank you so much.
Okay, so I'm gonna if I ever share any link, I'll try to share in Zoom as well, so that we are all on the same page. Slack is much better in those kind of situations, but I don't know what's happening now.
Only if you can see it though.
Yeah, true, true. It is. Yeah, indeed. Okay. I think I'm gonna start now. I think people are coming too as well. So yes, I mean, so basically, we're gonna use the group space, and it is better if you have a bootcamp folder under your name over there, and then you can go into bootcamp. And if you copied the files from the path that we have instructed, you should then have a folder called scrnaseq over there. So let's take a quick look at the content over there. I have some other folders over here because this is these are basically the folders associated to the Dolphin Next pipelines we're going to run. But what I'm expecting is that we have 3 folders over here. At the first folder, the PBMC1K3 fast queues are actually belong to the fast queues of the sample R1 and R2 reads. So we're going to slightly talk about the content of these too, to see what they are. Then we have two additional folders here. Right. So these are barcodes, features, and matrices, and we're going to talk about them. So these are basically the result of the single cell pipeline. They are, in a way, UMI tables, but we're going to actually try to understand how they entail UMI tables and how they store the UMI counts in a more efficient way. So what we're going to use as a tool is Cell Ranger. We're going to have Dolphin Next pipelines that we can use Cell Ranger. So let me quickly go to Dolphin Next as well. So all of you guys should have your Dolphin Next accounts now. And all of you should be able to find the, if you go to the pipelines in Dolphin Next, you should be able to have under single cell pipeline section single cell, you should have Cell Ranger pipeline. Just click on that. It's a little bit slow today. It's extremely slow like that.
Don't know. Yeah, it's sometimes getting slower.
Yeah.
Let me check if it's the network. Sometimes school network is
Yeah, I know.
not good, or everybody is connecting there now and making the machine slower. These are old machines, so we are moving everything to a new machine.
Yeah.
So it's gonna be better, hopefully. Yeah, I can. It's fast for me. I don't know. Still working for you?
No, maybe there's a problem with my connection.
Let me kill some people's runs on that machine. Let me see. Yeah, lots of people are using. Okay, what are you trying to show now?
I am trying to go to the Cell Ranger pipeline.
Just show the pictures now, and when it comes,
Yeah, sure.
don't wait,
Okay.
because we don't have time.
Okay.
It's not important to show it there now.
Okay, so basically, we can talk about Cell Ranger a little bit. It's a simple tool for aligning and quantifying single cell transcripts. The command that we're going to use, Cell Ranger count, is actually wrapped in one of our Dolphin Next pipelines, and that is basically the way that we pre-process a huge number of samples in the same time. You can go to what is Cell Ranger, website to learn more about the pipelines and how they are being used. They're basically using the same command, but what Dolphin Next is actually doing is that it's providing you an interactive platform to easily run Cell Ranger count. So there are multiple tools that we may actually incorporate in the same pipeline. Cell Ranger count is obviously the first one. And we can actually use Cell Ranger aggregate as well in the same Dolphin Next pipeline to aggregate multiple runs from multiple Cell Ranger counts. So it's a really nice website. You can click on each of them to learn more about them, how to run them. Specifically, the code is revealed explicitly, and there are clear instructions on how to run it. But of course, we usually prefer using Dolphin Next. So how we actually use Dolphin Next is that, maybe I can, briefly talk about the reads a little bit too. So you have two reads in the 10x genomics if you have certain chemistries. So we're going to use a diversion tree chemistry, which you can learn more about here in this link, and learn about the chemistry of the nucleotide sequences, and how much base pairs each barcode has, and the length of each UMI, and it's depending on the technology. And that will be one thing that we have to be careful of when we analyze this data in the pipeline. So basically, let me show it over here. So basically, you have compressed FASTQ files. Cell Ranger actually accepts the compressed versions of the FASTQ, so you don't really have to decompress them. But I compressed it for you so that we can take a look at each FASTQ file on them. So in each sample, you have two reads. And these two reads are actually different. The first one is a combination of the barcode and the UMI associated to one read of a transcript. So basically, if you just do a head -4 to get the first FASTQ read information on the FASTQ, you can actually see that these, sequencing, the sequence is actually of length 28, because the first 16 is belong to the barcode, and the remaining 12, which is associated with the third chemistry of 10x, is belonging to the UMI. And if you do the same thing with R2, you can actually see that this is actually the actual transcript that is associated to this UMI and this barcode. So basically, this means that this RNA is actually, this RNA read is actually belonging to this barcode and has this UMI. And if there are other reads with the same UMI somewhere, it means that it's actually the copied version of the same transcript. So how we're going to use this in Dolphin Next is that we're going to use the wrapped up version of our Cell Ranger command. I'm going to just try to see if I can open it, and I'm just going to stop trying if I can't. Permission denied. I don't know what's happening. Okay. Oh, there you go. Okay. So, so what's happening over here is that you have the reads defined as an input in this workflow. And once you have each of these pairs of each sample, and in this case, we're going to only have one sample, but it's going to be associated with two reads, which is the R1 and R2 over here, as we just talked about it. Over here, it will be used to run the Cell Ranger count command, and all the parameters are associated, are appropriately defined in the Dolphin Next pipeline, so that you can just go ahead and use it. Any parameter that you can use in Cell Ranger count command will be available to you in Dolphin Next as well. And you can use other parameters to do quality control on the FASTQ files, even initiate multi-QC to get additional results on your FASTQs as well. So, Alper, do you think we should allow people to try running, or shall we skip it? What do you think?
It's up to you. So you can, yeah, the people can run. Yeah, why not?
Okay, let's try. I mean, let's talk about it a little bit, right? So, so the only thing that you have to do in this case, by the way, guys, I must tell you, using Dolphin Next is super duper simple, and I must actually tell you honestly that the first time I learned how to use pipeline was through Dolphin Next. So it must actually tell you how it is actually easy to use, and I just shout out to Onur for creating this amazing tool with Alper. You know, it's basically much easier than using the terminal, and I'm grateful to them. You know, I use DolphinX every day. So you basically, if you have a new run, you have to go to, you just click run. It's just going to ask you if you want to create a new project for this run, or you can select any of the existing projects. So here I actually tried this, you know, this pipeline before, and I opened the project. But what you can do, you can create a new one saying test project, for example, and just save the changes, and it's going to create the project for you. Then you can just click on it and select the project, and it's now going to ask you what do you want to name your run. So you can just go ahead and say PBMC Cell Ranger test, or whatever you want to name it in this case, right? And this run will belong to this project, and we'll be able to use Cell Ranger right away. So there are some instructions in the GitHub page as well, if you're following. So I'm just going to, you know, quickly, you know, share it over here in Zoom so that you can see it. I'll open the chat. I'll put it over here too. Okay. So basically, you have to choose the reads first, right? So what we're gonna do is that we're gonna first define which of the directory in, you know, in your project space should actually have the actual DolphinX run. In this case, I have defined it over here, but I can just name it first. I can just give it a name, say I already have a PBMC Cell Ranger, but I'm going to just name it 2. So I just want to go over here to tell you what's going on. So the link over here should be the same as the link over here, right? So we are working under, you know, your group name, your username, bootcamp, and strnaseq. So you can open a directory for this specific run under this pad and just give any name to it. So I actually have a directory called PBMC Cell Ranger, but I'm just going to create Cell Ranger 2. So in this case, it's really easy. What you have to do, you just have to fill in these parameters that is required for the pipeline to be run. So you have to check pair because we have 2 reads, and perhaps it's, even though it's easy, the most complicated part over here is to choose which reads that you're gonna use in this case. So there are some additional instructions here in the GitHub page that how you can add reads to a run, but I'm just going to, you know, show that really briefly. So, okay, I have to choose the environment first. So if you have the UMass cluster environment set, you have to choose it. Then you have to add the read then. Then it's going to ask you the path, right? So where are the FASTQ files? It's here. It's going to just quickly check them, check the link, check the path. Just going to put it over here. Yeah, it's going to show you all the files under this path. It's FASTQ, of course. And I'm going to choose paired list because I want both R1 and R2 associated with the sample. So if I were to just click one of those, and as I just said, Cell Ranger actually accepts the compressed versions of FASTQ files. You don't really have to decompress yourself. So what you have to do, just add the selected files, and it's going to be added, and it's going to parse the name for you. So it's extremely easy. The only thing now that you have to do is to choose the collection name. And now this sample will be set for this collection. If you had multiple samples, you would be able to actually choose it from here and put it under the same collection. But for this case study, we only have one. Yeah, there you go. So I have a new collection. You have a sample over there. It's already set. And I have everything here. You can just click on the details and see if the path is right and everything is right. And just let's say. After this, you have to choose the genome reference. So we're going to use this latest HE38, gene code B32, cell ranger reference that we already have in our system here, exactly. But when we choose it from here in this list, it automatically adds the reference as a parameter where the path of the genome reference is exactly put it over here. The only other thing is that for this case study, as it was described over here, you have to define an additional parameter called accept cells 1000. So we're just going to do that. But DolphinX actually allows you to add these parameters because we already defined it. You can just change the 3000 to 1000 and click OK. In this case, we don't really need a FastQC to be run or aggregate libraries to be run. We're just going to leave it. So in this case, you have everything we said. You can see that the yellow button waiting is actually turned run, means that you are actually ready to run now. So I'm not going to run this myself. You can just click on it and it's going to show you that you can either start or manual run. You can just click on the start and the pipeline will start. Basically, this is a really good example because these pipeline will take 3 hours to run. So I'm not expecting that we're going to get the results now, but we are going to talk about the results in a different way. I already have a pipeline that I have the results. So I'm just going to go to that and we're going to discuss the report. So this is a version where after 3 hours, your pipeline is done working and this report section is popped up. And what you want to actually see is that you have this report that is in an HTML form is actually telling you how many of the cells are of high quality, have some number of UMIs that is higher than some threshold, how many genes per cell in average you have for each of these cells, and how many reads you have on average for each cell can be shown over here. You can actually take a look at the analysis. And the interesting thing is that Cell Ranger actually is giving you an initial analysis of the data, which clusters and do the dimensional reduction for you. Even though this is nice, you can see that there are some clusters, there are some cells that are clustered to each other, which may indicate that either these are T cells or NK cells or something like that, or populations like that. You can actually see it over here. But what we're going to cover, especially on Thursday, is that you want to actually do this thing manually, even though it actually helps you understand if your sample is good, because there's some good separation between the cells. Some of them could have a totally different phenotype than the others. It will be much more appealing to analyze it yourself, see the UMI counts. So in this case, this is kind of like a black box to you, but actually may help you understand if your sample is good or not. But the most important thing is the result of this analysis. And if you go to the folder associated to this run, so let's go to the settings to see where the run is actually located. So this is this path. I'm actually here. So I'm actually in this exact folder that DolphinX was run. I come over here and just check that there's a run folder and the report folder. And the results will be on the report folder. So on the report folder, you go to cell ranger count. You go to PBMC out. There's this separate folders over here. There is this raw feature BC metrics and there's this filtered feature BC metrics. Basically, these are all the barcodes that you have in your single cell experiments, regardless of the number of UMIs that you have captured in the experiment. But Cell Ranger actually filters some of those out, believing that they could belong to empty droplets, and gives you an additional folder with the filtered amount of cells in it that you can actually start with. And if you believe that you want to do your own filtering to a more advanced level, you can either use this folder, the outputs of this folder or this folder. It really doesn't really matter. Right. So, so these are the files that we're going to use, and these are the files that we're going to toss back in our local computers and analyzing R, which will be where we will be starting the actual data analysis. So if you go back to, is anybody not following, by the way? Am I going fast? Are we good? All right. I'm just going to go to the scRNA-seq folder in our bootcamp folder. Again, there you're going to see that there is this folder with the results of the Dolphin Next pipeline is actually here as well. So now what I'm asking you guys to do is to put this in your local computers by opening FileZilla. Where's my FileZilla? So I'm guessing everybody knows how to use FileZilla, right? Anybody who doesn't? Just raise your hand if you have any problems with using FileZilla. Okay, so Kashi, you can find the instructions in the first session, I believe. Yes. So this actually tells you how to download it and how to use the FileZilla in order to download the code. For those of you who are having problems using FileZilla and importing the counts, UMI counts, we're gonna have an alternative link from where you can use it from R. But if you can, just try using it. So basically, what we're expecting is that you can use FileZilla and you can import your. It's going to go to my desktop to show you what's happening. Import your result folder here. So basically what I'm asking is that you should have a folder in your local computer, just transfer it with FileZilla or any other client and have these three, files in it so that we can move on with the data analysis and use Surat in order to import the UMI tables from them. Just to quickly cover what these UMI tables, these files are, let me just go to the session to show you that. Okay, so this is where things are getting really, really interesting. Normally, when you're doing bulk RNA-seq, you only have, some few number of columns that are associated to your biological replicates, even though the row names will, let me go to that first example. We just take a look. The row names will be genes, which will be some 30,000 or 50,000, genes. But the column names will be some number like maybe 10, 20 based on your experiment. But it's never going to be around 10,000, 20,000 because in single cell, we have those kind of numbers. So basically, we have the same amount of RNA, but we have separated those RNA into cells of origin, right? So I'm expecting that we're going to have a lot of zeros in these expression profiles, right? So each expression profile will have a lot of zeros so that the important thing is that we shouldn't actually use all of them. And we shouldn't actually have a huge table with some column number of, 10,000, which all are zero. So instead, we're going to only store what is not zero, right? So what actually these three files are entailing is that you have a list of barcodes, you have a list of genes, and the metrics here is actually showing you which gene, which barcode of which gene is how many UMI. So basically, if you open the barcode list, you have the list of the barcodes over here in the barcodes file. In the feature file, you're gonna have the list of the genes over here. And what is actually shown in the metrics is that how many genes we have, how many cells we have in the first row, and how many UMIs in total across all the cells and across all the genes we have. And so how you have to interpret the second row below that is that the 17th gene in the gene list and the first barcode in the list has UMI count 1. So basically what's happening is that you have a joint table, but this is kind of like an aggregated row-based 3-column table, where it shows you this row name, this gene, and this column has number 1. So it basically allows you to not to store zeros, which actually allocates some space in your local computer, but just allows you to, store the non-zero ones, because we already know that all of other ones that are not zero are just 0. So we don't really have to store them. So the only thing that we have to do, we have to just put it over here in your desktop. And I also want you to open R now so that we can start analyzing. Is everything clear until now? Anyone who's not following? Okay. So the first thing I may want you to do, I want you to set your workspace in R to the desktop. So you can do that by clicking on the session up there and use set working directory and choose directory. Just click on it. It's going to open up a page. And if you dropped your PBMC process, this folder in your desktop, you're going to be able to see it. This is the place that we want to create our R script file, as Alper was mentioning in the previous 2 sessions. So I'm just going to click open. So now I'm in the desktop, right? So one thing that you can do to make sure that you're in the desktop, you can just click on the files on the right over here to see your files in the desktop, right? And the PBMC 1K version 3 process is over here. So let's start programming. Let's go back to the. So what we're going to start with is filtering. Single cell RNA-seq is a long process. You have to make sure first that your cells are of high quality, that you can start analyzing them, because it's going to be important for normalization. You don't want to normalize a cell which doesn't look like a cell, but it's actually perceptual. I mean, sometimes we have debates over what actually constitutes a cell, what is the appropriate number of UMIs in order to consider a barcode as a cell, to make sure that it's not a bad cell or it has high amount of UMIs to be analyzable. So it all depending on the technology that you use, or the efficiency of the library that you prep. So the library could be bad. The technology may not allow you to have enough UMIs. It's all depending. But at the end of the day, it's up to you to analyze the UMIs. So then we're going to normalize, and we're going to start choosing genes. Then we're going to drop the number of genes to some manageable number by machine learning dimensional reduction methods that Alper slightly covered in his talks, but we're going to go into much more detail on Thursday, and we're going to cluster as well. But today we're going to start with quality control. How we're going to analyze this data is that we're going to use the Seurat package. I briefly talked about it. The link is over here. You can see more about what the features of Seurat is, different kind of things that you can do with Seurat. You can just go to the link, just click the vignettes and just see some tutorials over here. So for different purposes, there are some advanced stuff that I'm not going to talk about either today or Thursday, but you can just go ahead and check, and their instructions are quite clear and immediately applicable. So you can just copy paste this, download the libraries appropriately, and start analyzing. And we're gonna kind of follow a similar progression in these tutorials as well. So what I want you to do is to first load the library. If it's a new script, you can just go ahead and say main 1K, give it a name, and we'll start analyzing. And we are still in the desktop, so we'll be able to import the folder that has our UME table in it as well. So the working directory is set. The only thing that we have to do is to run it. Let's see. It's going to quickly run Surat. This code will allow you to install Surat if you don't have it. If you have it, it's just going to skip it. So that's why we have an if statement over here. So this is the power of these kind of packages, guys. So when you have some kind of advanced UME table that is stored in a different way, but you don't really want to write your own code and just get the UME table right away, you can actually use the built-in functions that are in any kind of packages. The first one of these functions is red 10x, right? So I mean, just to make a suggestion, every time that you run a function, I would suggest that you actually type help. By the way, is my R studio big enough for that everybody can see it? Is this small? Is this looking good, Alper?
It's good. It's good.
Is it good?
Okay. I can read without glasses.
Okay, I see. Nice. Good. Okay. So guys, let's anytime that you have a package like this, you may actually analyze single cell with another package, even though Surat would be the most popular and the to-go package for a lot of people. Those packages have different functions. For example, we've seen DE-seek2, we have seen DE-browser, and Alper's DE-browser package and application has its own functions, right? But the amazing thing is that you can just use these functions right away, and you may actually look at instructions to see how you can actually code with them. So what I'm going to do first is that I'm going to write help read 10x and just get a help page of it. So the best thing about RStudio is that as soon as you click on help, you can see that instructions on how to use the read 10x function and the appropriate parameters, they are all here. So the description will say that it enables easy loading of sparse data matrices provided by 10x Genomics. The sparse data matrix that it's referring to is the file format we just talked about, right? This file over here. It's what we call a sparse matrix. A sparse matrix is a table that has a lot of zeros in it, right? So basically what this function is doing is that it allows you to read sparse data matrices whose files are appropriately defined in a folder to be quickly imported as a UME table. And you don't really have to code it yourself and turn it into a table yourself. So what I'm going to do, I'm just going to run it. You'll see that it's super duper easy to use. So now we have the object that actually has this. So now I just want you to remind you of some of the things that I want you to be really careful of. For example, never do this. Why is that? Because each function in R has a value, right? And that value has to be allocated to an object in R, right? If you just run this, it's going to give you the result, but it's not going to store it anywhere. I can just click on it. Now it's going to show me the results, going to show me some table, but that table will be nowhere to be found. So what I'm actually want to do is that I want to store the result of that function into an object that I'm going to reuse later. basically, I'm just naming this object that storing the UME table that I have into an object called PBMC1K, right? So you can just, type PBMC1K, click on it, it's going to show the same UME table. And the reason that we are not seeing any numbers over here is the reason that we have a lot of zeros, right? So we don't have many transcript coverage, there are some number of genes, but in most of these cells, a lot of counts will be actually zero, right? But some of them will be really, really high. For example, let's say that you have some T cells, the CD3 counts of those cells will be, I doubt that there will be zero, but they will be quite high, right? So the entire reason that we're doing single cell analysis is to understand which of those cells actually have non-zero counts in some of their genes and to identify those genes. So let's quickly take a look to every different thing that we can do with Surat. You can, it's written over here, but you can take a look at its dimensionality. The only thing that you have to do, you have to type dim, which is short for dimension, and you can type PBMC1K. All right. So another thing, which is really, really important for analysis in R, is that each object, here in this case, PBMC 1K is an object. In R, all objects are of a class. What do we mean by a class? So let's say that we have some arbitrary, value like X. Now X is an object, its value is 2, but you can actually check which class is of X. It's just some numeric number, right? But that's what it says. So X is a class of numeric, right? We know that it's a number, right? So we want to do the same thing to all different objects that we have. So what I can actually do in this case is that I can do class PBMC1K, right? So it's a matrix, right? But it's a sparse matrix because not all matrices are visualized like this. What we do in Surat is that we have a much more sparse version of the matrices in R, so that the size of the object will be much smaller because we have some 1,000 number of cells and some 1,000 number of genes that, well, not all computers may be able to handle it. So it's a nice way of storing the metrics, even though it looks like a matrix now. So moving on, you can actually take a look at different properties of this matrix. So I'm going to use a special package over here. So I'm going to use the same method. I'm going to check if matrix package is downloaded. It's not, it's going to install it. If it is installed, it's just going to pass and it's going to tell me that the library matrix is loaded. So now I'm going to use these NN0 function to understand how many zeros that we have. Basically, the specific function that I'm using over here is that PBMC1K is a matrix, and I use apply function to apply an N0 function to each of these columns over here. Basically, we have columns as barcodes or cells, and what we're trying to see is the number of non-zeros. So N0 is short for number of non-zeros. So what we're trying to achieve is the number of zeros in each expression profile, which is each column in our UME table. So when I run that, it's going to show me the entire list of zero counts that each barcode has, right? So basically what this says is that this barcode, which is associated to some cell, has 3,000 number of non-zero units. So I can actually guess that this is a really good sample because I was able to capture a lot of UMEs in it. So you can take a quickly summarize these things to see what are the different number of total UMEs or non-zero genes that you have in your expression profile. As you can see, the minimum number is 13. This means that one of the barcodes only have 13 genes, which is non-zero. So in this case, it will be up to you to determine if this is a good cell or not. or if you can work with it. Not all cells actually pass the filtering of Cell Ranger, even though it's filtering itself. So, I mean, it's again up to you to decide if you want to use the cell or not. So basically, you can, use other commands to see that almost 95% of all the counts in the matrix is 0. That will actually further proves that you have a lot of zeros in the UMI table. But you have to tabulize this eventually in R, because most of the analysis methods that we use in R are based on matrices. So in some way or another, they have to be written to some kind of a table. And those zeros should be there so that we can actually quantify them and analyze them. Okay, so the next thing that we're going to do, now we have a matrix, but we have to start analyzing with Surat. So what we're going to do is that we're going to create a Surat object out of it, right? So here, you can actually take a look at the help page over here too, to look a bit more detail about how we use create Surat object. So everything is object in R, and we use objects. And most packages, they have their own objects themselves. So in order to use the built-in functions in packages, we're just going to wrap those matrices or other data and tables into appropriate package-specific objects to start analyzing. So it's really easy to use. You just only have to define it. So I'm just going to change the object name a little bit. I'm just going to put a underscore after PBMC1K, short for Surat, to make sure that this is the Surat object version of this matrix. So you can just click on it. It's going to give you a brief summary of what is in the Surat object. So it's basically, it's just an empty object now. We didn't start analyzing in full. I have a question. Let's see. Is it that possible a cell has more than an mRNA? It's a good question. Well, I mean, theoretically, if you have that many, UMIs, so yes, but I mean, at the end of the day, it's a really high number to make sure that we have enough room to capture a lot of, reads in a barcode. But it's depend on the biology. So, I mean, we can say that it's maybe theoretically possible, but I've never seen it before. Did that answer your question, Oliver? All right. So moving on with the Surat object. So basically, the sparse matrix that we have is actually stored in this object. The components of the object is a lot in number. So there are multiple components that are stored in the Surat object. And what you actually can see is that you can actually approach them through, looking at different components. Say that you want to check if the count matrix is there, that we established the Surat object. You can just click on at. You can choose assays. You can click on $later, which give you RNA, but in this case, Surat is able to analyze multiple assays like attack-seq and maybe site-seq, or other kind of modalities that is available to analyze along with RNA. You can store them in the same object too. So this is basically the reason that we are using R objects, right? We are trying to simplify the process so that you don't really have to have multiple objects to track down your analysis, but Surat, well, in this case, will enable you to store each part of a project and put it in a same object that is accessible from the same object. So you can click on it, then you can click on the add again, and you can see the count metrics over here. So what's happening after we come up with this Surat object? So each single cell object, well, it's basically the case for a lot of single cell analysis tools and packages is that the UMI tables are accompanied by a metadata. So in order to get the metadata quickly in R, you can use the view function. The view function will allow you to view any kind of table, but we are going to use the metadata that is attached to PBMC1K Surat object. So in order to see that, you can use the same notation. You can start with @ right? But instead of choosing SA, you're going to choose metadata. The metadata is again a table, but instead of having rows as the genes and columns as the barcode, now the rows are the barcodes, and the metadata is actually telling you about additional features that each barcode has. In this case, since we just started analyzing this data, the metadata will only have some basic initial information about the cell. The first column will be the project name, where you can set it up in the beginning when you're using the create Surat object function. But then it's going to automatically calculate the total number of UMIs in all cells and the total number of non-zero genes, which will be named as features, because basically you can use the Surat object to analyze single cell ataxic assays too. So feature is a general name for each feature of a cell, which could be a gene or a part of a chromosome or anything that you want, right? It's based on the experiment. So other parts of the object are actually empty. We are just going to fill those out later, and we'll see how they are going to be used, but they are all in the same package. So let's start with the actual analysis, right? Let's see what we can do with it. The first thing to do is to understand if some of those cells are of bad quality, right? So you can use this function here to plot these features. So each features are actually parsed from the metadata. You can just put it over here, right? So what we're asking is that we want Surat to plot the UMI count of each cell and the number of non-zero genes of each cell in a violin plot. And the function for that is just VLN plot. And you just click on it and it's just going to give you the result right away. So how do we interpret this? So what I'm seeing is that mostly the cells have around maybe some 6,000, maybe 7,000 UMIs in total, right? Some of them slightly higher, but I can see that some cells over here, they have a lot of UMIs, right? So going back to dropsy, it is possible that you accidentally suspended multiple cells in the same droplet. So that's why the UMI count is so high. This actually happens in the analysis, and there is what we can do with Surat is just to assume those cells are not practical to analyze and just say, okay, they have a lot of UMIs. They are not really similar to all other cells. I'm just going to remove them later, right? You can just do that too. So what you can do, you can visualize N count and the total UMI and the number of genes in the same time. What I'm seeing is that there are cells that have extreme number of genes and extreme number of UMIs, making sure that those UMI counts are not coming from few number of cells. They're actually coming from a lot of genes, right? So this is further indication that I may have accidentally suspended 2 cells with different phenotypes in a droplet. Go ahead, Kashi. Maybe we can address questions later because we don't really have much time. Just going to try to cover. Then if you have any question, I can just answer it later. Is that okay with you?
I didn't ask anything, but.
Oh, you raised your hand.
Oh, I didn't bring it down. Sorry.
I'm sorry. Okay, I saw it. So I'm sorry. I thought that you did. Another quality control measure that we use in single cell analysis is the mitochondrial content. So basically, it is highly possible that when you're analyzing your cells, some of those cells may be going through a apoptotic state. When this happens, some of the RNA that you're going to capture is actually belonging to mitochondrial DNA, right? And that is actually captured when you isolate those cells in the droplet as well. So the thing that we are going to do now, we are just going to see what is the total number of those mitochondrial, UMIs, as opposed to the total UMI count in all cell to get the percentage of mitochondrial genes in your single cell object. So the way to do that is really simple. By the way, guys, now you should be able to understand is that all single cell related operations are super duper easy to use in R because it is well packaged in Surat and you can use them right away. So basically what this function is doing is that it's grabbing all the genes that are starting with MT dash to indicate that these are the UMIs that are captured associated to the mitochondrial DNA. And you get a fast percentage calculation on it. When you do that, we actually point this information to the metadata. So as you can see, the metadata will be changed. So in order to see that change, you can quickly view the PDMC1K Surat metadata. There you see. So now, along with the total UMI count and the number of genes, I actually have the percentage of MTs as well. So let's quickly draw a violin plot of that to see what could be additional filters that we may have in order to find out about the good cells. It's really interesting now that you should see some cells, even though they may have not many number of UMIs, most of those UMIs are actually coming from mtDNA, right? So, I mean, yes, they are apoptotic cells, they are dying, but they actually do not have any purpose in our analysis when we're trying to find out the phenotypes of T cells, maybe natural killer cells, right? So it's our duty to take a look at these violin plots and determine the best way of figuring out what could be the filters of those, right? And you can actually use the future scatter function again here and quickly run that as opposed to, let's do ncount RNA. Okay, as you can see that some number of cells have some considerable number of UMIs, but as I can see, as the percentage of mitochondrial content is increasing, their UMIs are quite low, meaning is that they have nothing other than mtDNA to contribute to the expression profile of a cell. So they are probably needed to be removed. And taking a quick look at these, chart, you can actually see that maybe, just maybe, somewhere around 20% is a good threshold, and you can implement this threshold to subset your Surat object. So I'm just going to quickly show how you can subset it, and we will going to stop today. You can use the subset function quickly to choose some of your thresholds, because let's say that we don't want to see cells that have some number of genes that we just saw that they are super low. We may not want to use them, right? So you can just say that I only want those cells that are higher than 20 to 200 genes, and some cells had more than 20,000 counts, and we don't really want them either. So you can implement this threshold, and of course we are just going to remove all those cells with higher than 20% as well. Let's take a quick look at the number of cells over here. It's around 1,200, but then I apply this new threshold and just point that to the same object. Now I'm going to just have around 1,000 cells. Basically, we have just removed some number of 150, 130 cells that we believe that were not quality. So I'm just going to skip the normalized data function now, because we can easily implement that. But for the record, we have another folder that has another example. That this example is a 3,000 cell version of the same biological sample, and your task will be to apply these quality filtering steps to analyze and normalize the data as this was instructed over here. But we will slightly cover these steps again on Thursday because we have much more room for tomorrow. So this is it from me today. If you have any questions after the lecture, I can take them. Otherwise, I'm good for now, and we're gonna move on with clustering and dimensionality reduction on Thursday. Any questions now? Before
I have a quick question about the mitochondrial counts. So is that the DNA, mitochondrial DNA, or mitochondrial RNA?
Oh, okay. So I might be using the wrong terms. They are mitochondrial DNA because at the end of the day we will have complementary DNA that we align to the genome. So we're just capturing those. So excuse me when I said RNA. I meant empty DNA.
No,
but what
no, it should be like, how come they can be DNA? Because we are doing RNA isolation. So these are coming from mitochondrial RNAs. Like,
So in a normal, like, let's say we have a normal healthy cell that's not undergoing apoptosis, and we're taking it through the 10x pipeline. And will you normally not expect it to contain any mitochondrial RNAs? Because I mean, it's a normal healthy cell, and so I'm assuming the mitochondrial RNA is doing something, some sort of function in the cell. So when you expect that during the RNA isolation process on the bead, and
no, but like if the cell is healthy, the mitochondrial RNA percentage is low. Like in the biology of cell death is doing that. It's increasing the number of, because the mitochondrial like cell membranes are getting just it becomes a mess there. So all mitochondrial RNA is going out from mitochondria. That's why it sequences more RNAs, I guess. I don't know. Biology better.
I got you. Okay, and I understand.
Yeah. So again, I mean, I'm not a biologist, but if there's something that we said not suit the terminology, yeah. But thanks for clarifying that, Alicia.
Yeah, but what we know about like single cell RNA seq analysis, when we see like more than 5%, 10% mitochondrial RNAs, we know that they are dead. So we want to remove those cells from our analysis. That's why we do this filtration before we start doing the rest of the analysis.
Just to clarify, Alper, they are dying. I mean,
they're dying, yeah.
Yeah,
dead or dying, yeah. They're not healthy. They're not healthy.
Yeah, I mean, they are no use. That's the bottom line.
Yeah, we understand. This is one way.
But Nazgul, to clarify your question, yes, there could be some subsets that you may isolate the mtRNA, right? But the most important thing is that whether they have additional RNA that have a high number of unique counts that could be of use. So let's say that you only have mtRNA or DNA, I just don't know which one to use at this moment, is that if you only have them, they are just dying. I mean, you have nothing to contribute. But it is possible that for some cases, especially for cancer, there could be some cells that have high number of those mtDNA that could be of use. What's most important is that what are those other genes that have high unique counts? Then they have high mtRNA or DNA. But yes, I mean, they can have additional phenotypes as well.
So during the lysis process, it's designed in such a way that the lysing only occurs, only the cell membrane is lysed to only capture the mRNA from the cytoplasm and not the RNA from the mitochondria, correct?
No. So if the cell membrane is compromised, the lysis can actually break down the RNA within the mitochondria as well. That's what I know.
No, during the 10x, there is a lysing step on purpose, right? To release the RNA from the cell, or no?
Oh, yes, it does, yes.
So that lysing step is designed in such a way that it only lyses the cell membrane, but not the mitochondrial RNA, correct?
But yes, but the cell is dying. It may as well actually.
No, if the cell is not dying, if it's a healthy cell, healthy normal cell.
Of course, yes, true.
Then the lysis buffer is such a buffer that it can only lyse the cell membrane, but not the mitochondrial membrane.
True, yes. So that is the indication that we have whether they are not dying or not. That is true. So it's correlated with that. So if it's a healthy cell, the lysis will only, damage the cell membrane, but it's not going to do anything to the mitochondrial membrane. But if it's dying, this is another case.
Okay,
As you just said, yes. Go ahead.
Yeah, so this is just to remind me that even just the regular RNA-seq analysis also consider this mitochondrial RNA.
Oh, so for that it's different. It's just that we have some cells that we are aligning to the entire genome along with all other RNA as well. So that this filtering is based on that. Just sure.
But I'll tell you something about the mitochondrial RNAs are also increasing in dead cells. There are papers you can check, like, mitochondrial gene levels in cell death. If you search like that, you're going to see some papers about it. They increase the number of mitochondrial genes. Maybe they are doing work, maybe even cell death requires more energy to kill the cell. It's just becomes a mess there. Just think about like that. Not only like mitochondrial cell membrane is going to be lysate, but also that the levels already higher, like mitochondrial gene levels are already higher. So you can think about like that.
Oh, so Alper, I totally agree. I believe the quality control measures that we implemented in this case are about that you're trying to align the entire genome of a healthy cell, that you believe that the cell population may have other cells that have no use for the functionality of mitochondrial RNA. So
No.
that's the call.
Unless you are working on mitochondrial genome, mitochondrial genes,
exactly.
Then we ignore them. Yansu, do you have a question?
Oh no, yeah, I'm okay then. Yeah.
You're good. Okay.
So
Thank you.
Kashi, I am not particularly well suited for talking about single nuclear RNA-seq, but we can talk.
Yes, just as an alternative, because sometimes we use frozen tissue. I was thinking about that.
Yeah. It is possible to get the RNA from the nuclei, but in that case, what happens? I don't know. Yes, I believe the mechanic will be much different. So again, I mean, the quality of filtering is highly dependent on the technology that you're using, and this mitochondrial, quality control measure is specific on that.
Yeah. So do you know the 10x single cell attack and RNA-seq, the kit that does both of them at the same time?
Oh, it's the new 10x multiome kit.
Yes.
It's using the single nuclei, yeah.
Yeah, so it uses single nuclei. That was why I was like.
Yeah, I see. So
Yeah.
I know. We're not going to touch base with multiomics in these lectures, but yes, I mean, the understanding is the same. All right.
Okay.
Any other questions?
Yeah. You can ask later too. Thanks for joining today's class. Thanks, Arthur.
Thank you.
Thank you.
Talk to you on Thursday, our last session. So come with your questions. Bye.
Bye-bye.